This paper was published in the journal Nature Communications in March 2016 and is titled Regulation of Cell-Cell Variability in Divergent Gene Expression. It explores the regulation of the expression of pairs of genes that are transcribed in diverging directions. This video will explain the findings of this primary research paper and is designed for students on the MGE course. First, let's look at some background information that will help us to understand the paper. So, what do the authors mean by divergent gene pairs? A gene pair is simply two genes that are close together on the same chromosome. If both genes 1 and 2 are transcribed in the same direction, they are said to be a tandem gene pair. If the direction of gene 2 is flipped, so it's transcribed in the direction of gene 1, this would be a convergent gene pair. And finally, if the two genes are transcribed in opposite directions going away from each other, they are said to be a divergent gene pair, or DGP. The authors of this paper focus on how the expression of this type of gene pair is regulated and how this can vary between cells. Now, let's move on to the methods the authors used and the results they collected. They start by studying divergent gene pairs and their promoters in yeast cells using the model organism Saccharomyces cerevisiae, or baker's yeast. They choose the divergent gene pair of the HTA2 and HTB2 genes, which encode two core histone subunits H2A and H2B. These two subunits always act as a heterodimer, so it is important that the ratio of the two proteins is regulated. Here is the structure of the HTA2-HTB2 divergent gene pair, with their shared promoter in the middle. When a transcription activator comes in and binds to the promoter, this induces the transcription of both genes, so this DGP is said to be co-regulated. To investigate the activity of this promoter in single cells, the authors create a transgenic strain of yeast with HTA2 replaced with a gene encoding the protein Venus, and HTB2 replaced with a gene encoding green fluorescent protein, or GFP. Now, when a transcription activator binds to the promoter, this induces the expression of Venus and GFP, which fluoresce yellow and green respectively under UV light. To assess the effect divergent gene pair structure has on expression, the authors also construct a strain of yeast where this gene pair is separated onto different chromosomes. They then record the brightness of each protein in individual yeast cells from both strains. While there is a positive correlation between Venus and GFP brightness in both strains, their covariance is significantly higher when they are divergently transcribed, which is displayed by a higher Pearson correlation coefficient, or R value, in the data from the divergent strain. This data supports the role of divergent gene pairs in coordinating the expression of two genes in a single cell. Next, the authors wanted to study differentially regulated GGPs, which are not activated by a shared transcription factor like HTA2 and HTB2. In eukaryotic genomes, most DGPs are differentially regulated. They choose the PFK26 MOB1 DGP, again in yeast. In times of heat shock, the transcription factor MSN24 binds to the PFK26 promoter, which activates its transcription, mediating a heat shock response. However, MOB1 does not play a role in heat shock response, so how is its transcription prevented in times of heat shock? Well, another protein, MCM1, associates downstream of the PFK26 promoter to prevent MOB1 transcription. To determine how effective MCM1 is at blocking transcription activation, the authors replace MOB1 with Venus. If Venus expression is successfully blocked during times of heat shock, there should be no yellow fluorescence in this yeast strain. They take this yeast strain, induce heat shock, and measure the intensity of Venus brightness over the next few hours. In typical yeast cells, shown here in light blue traces, Venus intensity remains low and stable for around 2 hours as the cells adapt to heat shock, before starting to rise thereafter. However, there are some outliers, shown here in dark blue traces. These cells show a significant increase in Venus brightness during the first 2 hours. The authors plot the slope of increase in Venus brightness in this histogram, which shows that although most cells do not express Venus in the first 2 hours after heat shock, a small fraction of cells do. This leads the authors to suggest that a leakage between differentially regulated DGPs occurs in about 15% of cells. In the final part of the paper, the authors determine the conservation of DGPs in evolution. They compare the genome of Saccharomyces cerevisiae to that of the closely related Clivoromyces welti. At some point after they became separate species, Saccharomyces underwent a whole genome duplication event, while Clivoromyces did not. The whole genome duplication dramatically altered the Saccharomyces genome organization through duplication and deletion, which provides the unique opportunity to study the evolution of gene order. The authors expect gene orders that enhance cell fitness to be more likely to be conserved, and vice versa. For every DGP in Clivoromyces, they bioinformatically determine whether the gene order has been conserved or lost during evolution in Saccharomyces. They find that the DGP structure seen in Clivoromyces is conserved in Saccharomyces in 454 cases, while 696 pairs are lost in evolution. They also find 700 new DGPs that were not present in Clivoromyces. They next perform microarrays to determine the expression level of each DGP. This allows them to calculate a co-expression score, which shows how closely linked the expression of each gene in the pair is. A co-expression score below 1.5 indicates that the genes in the divergent pair are differentially regulated, while DGPs with an expression score above 3 are co-regulated by shared transcription activators. This graph shows that differentially regulated DGPs are significantly less conserved than average, which is demonstrated by the high bars in green at low co-expression scores. On the other hand, the average co-expression score of conserved DGPs is higher than average, demonstrated by the taller orange bars to the right of the graph. Of the 12 conserved DGPs with a co-expression score above 3, 8 encode histone or ribosome units, indicating that there is an advantage in maintaining the stoichiometry of these proteins and thus the DGP order. The authors conclude that the high covariance of co-regulated DGPs is beneficial for cell fitness, and therefore they tend to be conserved. Conversely, leakage between differentially regulated DGPs may be harmful, and therefore they are minimized by natural selection. Finally, let's recap on the findings of the paper. First, we saw how the structure of DGPs allows the coordination of genes that need to be expressed at the same time via co-regulation. Next, we explored how leakage can occur between the two genes in differentially regulated DGPs. And lastly, we learned that co-regulated DGPs are more likely to be conserved in evolution than those which are differentially regulated. Thank you for watching this video. I hope you have found it informative. If you have any questions, please leave them in the comments and I will do my best to answer them.